Hello, I'm Jong-seo, and I wrote the article named Control of Foxp3 Induction and Maintenance by Sequential Histone Acetylation and DNA Demethylation. This article was published in December 2021, and the name of the journal is Cell Reports, which impact factor is about 10. Before starting the review, I'll show you a brief summary of this paper for better understanding. In this paper, they want to know the stage-specific regulatory mechanisms of Foxp3 in regulatory T cell. The answer of this question can be summarized in 3 sentences. There are 3 stages of the regulatory T cell, and they revealed the specific mechanisms of them. Then, what is Foxp3 and what is regulatory T cell? Regulatory T cell is a type of the CD4 plus T cell which are differentiated from naive CD4 T cells, which is a precursor. Most of the naive CD4 T cells are differentiated into helper T cell like Th1, Th2, and Th17. Only some of the naive CD4 T cells become regulatory T cell. Regulatory T cells are essential for our immune system. They maintain immune tolerance and homeostasis of the immune system. It means that they regulate or suppress other immune cells, and they control the immune response to self and foreign particles and help prevent autoimmune disease. Then what is Foxp3? Regulatory T cells express IL-2 receptor alpha chain, and also they express high levels of transcriptional factor Foxp3. Foxp3 deletion in the germline lead to improved development of Treg cell. Foxp3 overexpression in other CD4 plus T cells gives rise to a Treg cell-like fate. Therefore, Foxp3 is essential for differentiation into Treg cell and maintaining the identity of the Treg cell. So they want to know how this important gene is regulated in this cell. In the stage of initiation, histone acetylation of Foxp3 promoter in cis activates Foxp3 expression. In the stage of transition, histone acetylation in trans activates Foxp3 expression. After differentiation into regular T cell, hypomethylation in CNS2, which is Foxp3 enhancer, makes stable Foxp3 activation. Before starts the main results, I tell you what is the term cis and trans. Cis and trans are terms for transcriptional regulation method. To put it simply, cis regulates gene expression by direct binding. In contrast, trans means that the global regulation by the abundance of certain protein after transcription. Then, this slide shows the first result of this paper. My explanation structure will be the main title of the result, and explanation of the background, key terms, and result or conclusion of each figure shown in the red box. The first title of the result is histone acetylation is required for Foxp3 induction in cis. In the beginning, they want to see the histone acetylation near the Foxp3 region before and after differentiation into in vivo natural regulatory T cell. The histone acetylation is previously reported factor in regulatory T cell development. That's why they want to see this signal. So Tn means naive CD4 plus T cell, which is precursor cell before differentiation. Anti-reg means regulatory T cell in vivo, which means after differentiation. You can see the acetylation signal in Treg at Foxp3 region, but not Tn. Figure B and C shows the number of the Foxp3 expressing cells shown in percent Foxp3 plus after treating JQ1 and C646. In this experiment, they use iTreg cells, which become regulatory T cell in vitro. C646 and JQ1 suppress histone acetylation pathway, and after blocking the histone acetylation by them, the number of Foxp3 expressing cell decreased. And figure D, the Foxp3 expression also decreased, which has high correlation with Foxp3 expressing cell ratio. Next, they want to confirm the effect of those inhibitors. So they harvested cells 24 hours later at the early phase of iTreg cell induction and undergo chip sequence to fully reveal the effects of these inhibitors. Figure E shows that the amounts of acetylation in the Foxp3 region were decreased when the inhibitor JQ1 or C646 was treated. And figure F shows the decreased expression of Foxp3. In figure G, you can see that the high amount of black signal was decreased after treating. So they found that histone acetylation in early phase of iTreg cell induction was decreased when the inhibitor JQ1 or C646 was treated. This part revealed that the histone acetylation induces Foxp3 in cis mechanism. In figure H, they treated JQ1 at 48-hour iTreg cells, although there is no direct explanation about the meaning of 48 hours. In this paper, I think the time 48 hours means that the initiation was already done and they are differentiating. And 24 hours means that this time is about the point of initiation. Then I thought that if the histone acetylation mechanism was controlled by trans mechanism, the Foxp expression after treating JQ1 will not significantly decreases because they do not decreases the amount of the histones. But this figure shows that there is significant decrease of Foxp3 expressing cell in JQ1 condition. And in figure I, they saw significant decrease of mRNA expression near the Foxp3 region at the start point of differentiation. This result supports that the histone acetylation is required for Foxp3 induction in cis. And please note that because this explanation is in my understanding, so it can be wrong. And the name of the next result is histone acetylation signal via BRD protein targeted by JQ1 is dispensable for stable Foxp3 expression in ASC-treated I-Treg cells. This means that the major mechanism regulating Foxp3 expression was changed after initiation and become stable. They started to check the methylation status because previously research reported that TET-dependent DNA demethylation was proposed as the main mechanism acting in cis to lock in Foxp3 expression in committed Treg cells. In this part, they made an in vitro model using differentiating iTreg cells by supplementing the culture medium with ASC that activates TET enzymes and induce active DNA demethylation. They made this in vitro model because in vivo experiments are not readily accessible. This model is shown in figure A. To assess the stability of Foxp3 expression, they sorted GFP plus iTreg cells both later and cultured them in media containing recombinant IL-2, AIL-2, or JQ1 together with immobilized anti-CD3 and anti-CD28 antibodies. If you just keep in your mind that AIL-2 provides unstable condition, you can understand the result easier. In figure C, you can just see the number in this figure. With treating AIL-2, mock-treated iTreg cells lost about 80% of the Foxp3 expression, but more than 80% of ASC-treated iTreg cells remained Foxp3 regardless of the culture condition, resembling stable anti-Reg cells in our body. These results suggest that ASC treatment separates the mechanisms controlling the stability and levels of Foxp3 expression. To further understand Foxp3 transcription during this transition stage, they compared the transcriptional activity of Foxp3 in mock and ASC-treated iTreg cells with or without IL-2 deprivation. In figure E, in AIL condition, slightly reduced Foxp3 transcription in mock-treated iTreg cells. It suggests an accumulative effect of weakened IL-2 signaling on the stability and levels of Foxp3 expression. In contrast, ASC treatment drastically increased the transcriptional activity of Foxp3, suggesting a sudden change of the mechanisms governing Foxp3 transcription. Then, they tested whether histone acetylation plays a role in maintaining Foxp3 expression in differentiated iTreg cells. They cultured mock and ASC-treated iTreg cells in media containing titrated amount of JQ1. In figure F, JQ1 silenced Foxp3 expression in mock-treated iTreg cells. However, Foxp3 expression was maintained in more than 70% of ASC-treated live iTreg cells. In figure G, JQ1 also decreased the expression. Therefore, histone acetylation signal via BRD proteins targeted by JQ1 is required to sustain Foxp3 expression prior to ASC-induced epigenetic changes. To test the requirement for TET enzymes, they removed TET1, TET2, and TET3 in CD4 N cells with retroviral Cre. In figure I, Cre-transduced iTreg cells significantly decreased the stability of Foxp3 expression in ASC-treated iTreg cells, shown in green, after JQ1 treatment or upon IL-2 deprivation. And in figure J shows a constant result. There is profound downregulation of Foxp3 transcription in Cre transduced iTreg cells regardless of the culture condition. So it needs TET enzyme. Put together, TET-dependent DNA demethylation switches the driver of Foxp3 transcription, conferring stable iTreg cell identity in adverse environments. They thought that histone acetylation signal may maintain Foxp3 transcription in cis or in trans. So they firstly checked H3K27AC levels at the Foxp3 promoter in iTreg cells. In figure A, they induced iTreg cells and 4 days later they treated them with or without JQ1 and labeled with SU. Analysis was progressed after 5 hours. In figure B is a PCA plot showing that this protocol failed to avoid the influence of JQ1 on total RNA transcripts. However, in figure C, they explained that there is a striking effect of acute JQ1 treatment on global transcriptional activities. In figure C and D, we can know that ASC treatment affected the levels of both total and newly synthesized RNAs. Then they checked the effect of JQ1 on new RNAs. Figure E shows that Foxp3 new transcripts were not significantly changed in JQ1 treated iTreg cells without ASC. Please remind that previous result showed that lower Foxp3 expression after long-term JQ1 treatment. In this short-term 5 hours treatment, Foxp3 used transcripts were not significantly changed. So long-term effects suggest that it is regulated by trans mechanism. Put together, this result suggests that histone acetylation signal is not required for maintenance of Foxp3 expression in cis, in differentiated iTreg cells. To understand how JQ1 destabilizes Foxp3 expression in the long term, they checked genes which are downregulated So let's keep going. Also, to understand how JQ1 destabilize Foxp3 expression in long term, they check genes which are downregulated in JQ1 treated iTRX cells. In figure H, they found several non-positive regulators of Foxp3 expression were drastically downregulated in JQ1 treated iTRX cells. So, histone acetylation signal via BRD proteins targeted by JQ1 maintains Foxp3 expression in MOC-treated iTRX cells in trans. Meanwhile, the data with ASC is not a main subject of this session because JQ1 treatment did not impair the stability of Foxp3 expression in ASC-treated iTRX cells. So please consider that this conclusion only valid in MOC-treated iTRX cells. The title of result 4 is CNS2 is required for TET and DNA demethylation dependent stabilization of Foxp3 expression. So to understand how ASC stabilize Foxp3 expression, we first find mapped differential DNA methylation sites in several T cells. In figure B, NTRAC cells were hypomethylated at Foxp3 promoter and CNS2 locus. We can see that ASC treated iTRAC cells exhibit similar hypomethylation with NTRAC cells. Based on this data and published reports, also suggested that ASC treated iTRAC cells is an excellent ex vivo model in which to study DNA demethylation dependent control of Foxp3 expression. To verify the requirement for TET enzymes in ASC-induced hypomethylation of CNS2, they removed TET1 to TET3 with retroviral Cre, and they saw that the effect of ASC on CNS2 demethylation was abolished in Cre. So, this result supports the reported low role of ASC and TET enzymes. To test the sufficiency of CNS2 for ASC-induced stabilization of Foxp3 expression, they examined Foxp3 expression in CNS2 sufficient or CNS2 deficient iTRAC cells. In MOC-treated iTRAC cells, TCR restimulation alone in figure D or in combination with JQ1 in figure E diminished Foxp3 cells in both CNS sufficient or insufficient region. Comparing these two, CNS2 deficient iTRAC cells appear to be less stable than that in CNS2 sufficient iTRAC cells, and these results suggest a minor role of CNS2 in maintaining Foxp3 expression before DNA demethylation. Besides, ASC-treated stabilized Foxp3 expression in wild-type iTRAC cells regardless of culture condition. In CNS insufficient iTRAC cells, it slightly stabilized Foxp3 expression. It is because additional genetic elements may also be involved. Put together, CNS2 contributes to most ASC-dependent stabilization of Foxp3 expression. The title of fifth result is DNA methylation controls chromatin accessibility and nuclear factor binding at CNS2. they want to understand how CNS2 demethylation controls Foxp3 expression. In figure A and B, they checked H3K27AC levels and observed a significant increase between Foxp3 promoter and CNS2 upon ASC treatment. In figure A and C, they assessed chromatin architecture with ataxic. Upon ASC treatment, there are 3,320 genetic elements with significantly changed accessibility. CNS2 also became accessible. And they assumed that this may facilitate nuclear factor binding. So they assessed STAT5 binding, which is a known factor of Foxp3 expression. In figure D, 39 elements were enhanced or weakened, and particularly STAT5 binding at CNS2 was drastically increased upon ASC treatment. ASC also increased RNA polymerase II occupancy at 117 genes with Foxp3 on the top. This is the minor figure. Figure F and G shows the correlation between increasing binding of STAT5 and polymerase II with reduced methylation and increased histone acetylation. To test whether STAT5 binding at CNS2 was Treg cell specific or not, they compared MOC and ASC treated Th0 cells and iTreg cells. So figure A shows that there is STAT5 binding at CNS2 in ASC treated iTreg, but relatively not in MOC treated iTreg cells, or ASC treated Th0 cells. In figure I, they also removed TET enzymes with Cre, and then the STAT5 binding at CNS2 was eliminated. It supports the role of TET-induced DNA demethylation. They thought that increased CNS2 accessibility may also facilitate the binding of other nuclear factors. They then examined Foxp3 association with CNS2, a feedforward loop proposed to stabilize Treg cell fate. It really increased in ASC-treated iTreg cells, but not in cells transduced with retroviral Cre. Taken together, although there might be more factors that differentially bind at CNS2, DNA demethylation controls chromatin accessibility and nuclear factor binding at CNS2. In result 6, they tested whether Foxp3 binding at CNS2 provides a strong feedforward loop in stabilizing Foxp3 transcription. In figure A, they used dye 1.1 to report the Foxp3 expression, but block the feedforward loop. In figure B, they generated chimeric mice which prevents autoimmune disease, and the dye 1.1 expressing wannabe iTreg cells was induced. Firstly, they checked the stability of dye 1.1 expression in vitro. In figure C, more than 5 55% of control iTrex cells and more than 80% of wannabe iTrex cells failed to maintain Foxp3 or Di 1.1 expression. This result suggests the role of the Foxp3 feed-forward loop in sustaining Foxp3 transcription through DNA demethylation and CNS-independent mechanism. Consistently, seeing the black bar in figure D, ASC treatment significantly enhanced nascent Di 1.1 transcripts. To examine the stability of Di 1.1 expression in vivo, they sorted their wannabe iTrex cells which present Di 1.1 and co-transferred them into Rec1 negative mice and induced iTrex cells. So in figure F, 2 weeks later, approximately 20% of mock-treated and 80% of ASC-treated wannabe iTrex cells maintained Di 1.1 expression in here. Next, they tested the stability of Di 1.1 expression in wannabe Trex cells developed in vivo. They sorted anti-Trex cells and wannabe Trex cells and co-transferred them into Rec1 negative mice. 2 weeks later, figure I shows that approximately 90% of anti-Trex cells maintained Foxp3 expression shown in black circle, and 70 to 80% of wannabe Trex cells remained Di 1.1 expression shown in blue circle. These results indicate that Foxp3's feed-forward regulation is a minor contributor to TET-induced stable Foxp3 transcription. In summary, STAT5 and Foxp3 binding as CNS2 may play only a minor role in stabilizing Foxp3 transcription in iTrex cells upon TET-induced DNA demethylation. In result 7, they additionally tested whether several known nuclear factors contribute to stable Foxp3 expression in ASC-treated iTrex cells. They knocked down these factors using CRISPR approach and found that it impaired the stability of Foxp3 expression in mock-treated iTrex cells. They also found that ASC treatment always has an additive effect on the stability and levels of Foxp3 expression, and which can suggest multiple independent pathways governing the stability of Foxp3 expression. This is the last slide, and it provides brief summary figure. Taking this opportunity, I'd like to remind the slide which I introduced earlier. They revealed stage-specific epigenetic regulation governing iTrex cells. First, in the stage of initiation, histone acetylation of Foxp3 promoter in cis activates Foxp3 expression, and it is inhibited by JQ1. In the stage of transition, histone acetylation in trans activates Foxp3 expression, and actually most mock-treated iTrex cells stays in this transition stage. After differentiation into regular T cell, hypomethylation in CNS2 with multiple independent factors and pathways make stable Foxp3 activation. In this stage, they used anti-reg cells with in vivo model, and they used ASO-treated anti-reg cells with in vitro model. However, they mentioned about their limitation that these two models may have considerable differences, but they used in vitro model. So this study should be verified using in vivo model. This is end of the presentation. Thank you.